Peptides are short chains of amino acids where the carboxyl group of one amino acid is joined to the amino group of the next amino acid. So we can break that down here with this example where we have two separate amino acids. So we can draw a line sort of right here. This red line down the middle is going to differentiate between amino acid, I'll just do AA to represent that, amino acid 1, and then AA2. So there's a lot going on here, but really we can break it down into the pieces that we know well. So let's just focus on amino acid 1 first. So we have our alpha carbon here, we have our amino group right here, we have a hydrogen right here, we have our R group, which is just the side chain. It can be anything. In glycine, it would be hydrogen. So it really depends on what it is. And then we have our carboxylic acid right here. So I don't want to draw the other stuff in yet. Or you know what? Yeah, that's fine. Carboxylic acid. Okay, cool. Same over here. In amino acid 2, we have all the regular things we're used to. So here's our amino group, alpha carbon, carboxylic acid, hydrogen, and then a second R group for, or rather an R group that is for amino acid 2, which we are notating as R2. So we have all those different things. I can delete all that just so we can get back to our original as well. The reason for that is because I don't want to overwhelm with too many things going on at once. So, cool. Let me just draw the 2 back. Okay, so here we're back to our, what we're calling peptide. So we have 2 amino acids, but we probably noticed as I was walking through each amino acid that the carboxylic acid and the amino group of both are different than usual. So we can see a typical one for an amino acid at physiological conditions here and here, where the amino group is charged and then the carboxylic acid group is also charged. And if we notice the carboxylic acid group of amino acid 1 and the amino group of, or rather, yeah, the amino group of amino acid 2 are both uncharged, and they're different because the carboxylic acid of the amino acid 1 only has 1 oxygen, and the amino group of amino acid 2 only has 1 hydrogen. And the reason for that is because when a peptide forms, the carboxylic acid of amino acid 1 loses an oxygen, and the amino group of the amino acid 2 loses 2 hydrogens. And what's cool is if we really think about it, if you're losing 1 oxygen and 2 hydrogens, you're really just going to be losing a water in total. And that's exactly what happens. There's the dehydration where the water gets lost, and something called a peptide bond is formed through that process. The peptide bond is going to form between the carbon of the carboxylic acid for amino acid number 1, and then the nitrogen of the amino group of amino acid number 2. And this bond, this peptide bond, is a very strong bond. This bond has shared partial double bond character, because basically if you think about this double bond right here between the carbon and oxygen, it's actually going to have sort of shared double bond character over the carbon and nitrogen here. And some other cool things to note are that this yellow portion that's dotted out around these 6 atoms are actually going to exist in the same plane, meaning that all 6 of these atoms are going to be, like if you lined them up on a piece of paper and the rest of the atoms are spread out across the plane of the paper, behind it and above it, these 6 atoms are going to be within that plane of the paper. Peptides are not just 2 amino acids necessarily. There can be polypeptides with a lot more amino acids, and then these are going to be the building blocks of proteins, which we'll talk about tomorrow.